All right, well, today I would like to welcome Dr. Morgan Zumbaugh, Assistant Professor in the Animal Science and Industry in the College of Agriculture here at K-State. Dr. Zumbaugh was a recipient of the 2021 Global Food System Seed Grant Award, and the title of her presentation is Understanding Skeletal Muscle Metabolism as a Means to Improve Livestock Production Efficiency. Dr. Zumbaugh?
Thank you for that introduction, and thank you for the opportunity to present our work. We're really excited about some of our findings and where this will take us in the future. So just a general overview, kind of the work that we've been able to do as a result of this seed grant is start to investigate the metabolism of skeletal muscle to use that information to ultimately provide innovative ways that we can improve feed efficiency of livestock. Technical difficulties. There we go. So if we look at the meat industry in the state of Kansas, I think it's pretty obvious the importance of the meat industry and just how large of an impact the meat industry has on the Kansas economy. And if we kind of look at this from a snapshot, you can see that the meat industry was responsible for about $13 billion of economic activity in the state in 2020. And if we look at this as the inventory of animals that we have in the state, you can see that there's about almost 13 billion animals that we have in the state of Kansas. And so this includes cattle, goats, sheep, and hogs. And these animals are consuming quite a large amount of feed. So when it comes to being able to improve the impact that the meat industry has on the state, what we're trying to do is figure out ways that we can make these animals more efficient in feed efficiency. So these animals that are consuming the feed, how can we make them more efficient and be able to use this feed more efficiently to produce meat? And so if we break this down by animals, you can see that when we talk about feed efficiency, so feed efficiency would be the ability of these animals to take that feed and convert it to a pound of carcass weight. And if we look at pork specifically, you can see that there's room for improvement when we talk about feed efficiency of pork. And so for the rest of this presentation, we'll actually be focusing in on pork as a species. And so if we look at the pork industry in Kansas, breaking this down, you can see that the pork industry has a market value of almost $500 million annually, and the state produces 600 billion pounds of pork. So not only is this of huge importance to the state of Kansas and our economy, but also feeding the country. And if we look at the feed costs that producers take on to produce this meat, you can see that Kansas pork production requires over 30 million bushels of grain, and this is on an annual basis. And so this alone is costing producers about $90 million, which has a huge impact on pork producers' bottom line. So what we're trying to do is determine ways that we can reduce the feed requirements of growing pigs in the state of Kansas. And if we look at historically in the United States and in Kansas, the industry has done a great job of reducing feed requirements. Over the past 40 to 50 years, we've had great advancements in genetics, nutrition, feed additives to be able to grow the same amount of meat with less feed. And this is highlighted nicely here, where we can see that from 1992 to 1998, there was almost a 27% decrease in feed conversion ratio, meaning that we needed less feed to produce the same amount of meat. However, if we look at this change from 2004 to 2009, you can see that there was only a 3% decrease. So while we've made huge advancements in the past, and these advancements are still just as important to producing meat today and in the future, we need to come up with innovative ways to continue to reduce the amount of feed that's required to produce meat in the state of Kansas and across the country. And so this leads us to kind of what we're doing here, and we're trying to come up with innovative ways to improve feed efficiency. And so this brings us to the question of, how do we do this? Where are we able to intervene and continue to make advancements? And so if we look at the growing pig and the tissues that are present in this animal, and we break it down by major tissues, we can break this down into vital organs, bones, adipose tissue, and skeletal muscle. And quite apparent, we're not going to want to interfere with vital organs or bone growth, because those are very important to animal function, animal health, animal welfare. So that leaves us with adipose tissue or skeletal muscle. And over the past 40 to 50 years, the industry has done a great job of following consumer demands of wanting a more lean product and less fat in their pork products. And so the industry has done a great job of kind of making interventions with genetics to be able to create leaner pigs. And so that really leaves us with skeletal muscle. Skeletal muscle has the ability to make significant advancements in the future. And so I think this slide does a good job of displaying that. So what we're trying to do is when animals consume feed, that feed is broken down in the digestive tract to the most basic nutrients. So carbohydrates, fats, amino acids, and the muscle is going to be exposed to these nutrients. And so when the muscle gets glucose, for example, and it takes up that glucose, the muscle cell is going to metabolize that glucose, and it's either going to use it to support some energy function like muscle contraction, or it can be used to support growth or muscle hypertrophy of this tissue. So what we're trying to do is figure out how muscle responds to these nutrients and how it kind of determines where those nutrients are going to go, whether it's going to go to being waste, lost to some sort of metabolic waste, or whether it's going to be used to support an increase in tissue mass. So kind of bringing this back full circle, by understanding these processes and understanding the metabolism of skeletal muscle, if we're able to improve the efficiency of muscle being able to take up these nutrients and use it to build more protein or increase the muscle mass, if we can decrease some of the inefficiencies that are naturally occurring in the muscle by about 20%, this would translate to about $13 million a year of savings, because less feed would be required to generate the same amount of muscle tissue. And so how are we doing this? If we take a look at how pigs grow, we know that muscle growth, we see large amounts of muscle growth kind of in the middle section of the animal's life here. And so what we're trying to do is determine, one, does the metabolism of muscle change with age? And if it does, what are these changes? And then how can we use these changes to then design intervention strategies? So whether that's genetic selection markers or different nutritional advancements. So we're trying to go from the cellular level up here of defining what's happening at a cellular level and how we're able to use that information to make changes on the whole animal level. So in this study, we isolated muscles from growing pigs. So we had pigs ranging from weaning all the way to market weight. And we selected various ages through this process as well. And so we collected 3 muscles from the pigs, and we collected the longissimus dorsi, which is a glycolytic muscle, the latissimus dorsi, which is a mixed muscle, and the masseter, which is an oxidative muscle. And so we selected these 3 different types of muscles because they inherently have different types of metabolism. So muscle as a whole is not a homogeneous tissue. There's different types of metabolism that are used by muscles depending on their distribution through the carcass. And so we've selected these kind of as our extremes and then as an intermediate to give us an idea of how the metabolism is going to change in growing pigs, and also how the metabolism might change when muscles have different types of inherent metabolism. So the objective of this study was to uncover mechanisms that determine how the muscle is going to allocate nutrients to these different metabolic pathways, and then use this information to develop strategies to continue to improve feed efficiency in the future. And I'd like to point out the picture here. So this is Linnea Rimmer. She is the graduate student that has been able to coordinate and lead this research project. This was when she was collecting samples. So after we collected the muscles, we then determined the abundance of key metabolic enzymes in the metabolism of muscle. And so this image here kind of shows us an overview of the pathway that we're looking at. And as we step through the data, we're going to kind of walk through these metabolic pathways. So the first part that we're looking at here is are these glycolytic enzymes. And so we've identified two key enzymes in glycolysis that have the ability to take metabolites and use them in anabolic or building pathways. So these enzymes here are highlighted in green because we already know in the field of muscle biology that these enzymes are associated with pigs that are more feed efficient. So we're trying to determine how these enzymes change as the animal's growing as an indicator of anabolic or building pathways. On the other hand, we've also looked at two key oxidative enzymes. And so these I have here in red, as well as kind of as we go through this presentation, because we know that pigs that have lower feed efficiency tend to have a greater expression of these oxidative enzymes, or enzymes that direct metabolites towards wasting. They get lost as metabolic waste or are used less efficiently. And then the third category that we've looked at is an enzyme that has been shown to direct nutrients or metabolites out of this catabolic pathway into amino acid synthesis. So actually synthesizing proteins to support lean growth. And so when we dive into this, we're first going to start with those glycolytic enzyme pathways. So G6-PDH is an enzyme that, again, is going to direct the metabolites into a building or anabolic pathway. And as we go through this, we again have the LD, which is a glycolytic muscle, all the way down to the MS, which is an oxidative muscle, and then this mixed muscle in between. And we looked at these again across ages. So what I'd like to draw your attention to here is in the MS, this oxidative muscle, you can see that between 104 and 137 days of age, the MS has a lower expression or abundance of G6-PDH compared to the LD or the LAT. And so what this tells us is that in muscles that are more oxidative at this time point, we don't see an increase in this building pathway. If we continue to look at another glycolytic enzyme, LDH, we can see here that the oxidative muscle, not surprisingly, has a lower expression of this glycolytic enzyme than the LD or the LAT. But what I'd like to draw your attention to is that in days of age, between 37 days, so weaning, to 104 days, about halfway through this animal's life, we see a decrease in LDH, or this enzyme that's able to direct nutrients into this building pathway. And as we continue to follow pyruvate through this pathway into the oxidative pathway, we can see that the MS, or this oxidative muscle, has a greater expression of PDH than both the LAT or the LD. So we see this kind of stepwise increase as muscles become more oxidative. But I'd like to draw your attention to 137 days of age. You can see that there is this quite large increase, almost a threefold increase in the abundance of PDH from 69 days to 137 days of age. And as we continue to step through this, you'll see this pattern kind of arise where we see a lot of enzymatic changes occurring from after weaning up until this 137 days of age. And we see something very similar here with citrate synthase, and this is that second enzyme in this pathway where metabolites are going to enter this oxidative or burning pathway. And we see the same trends across muscles. So as muscles become more oxidative, we see an increase in the abundance of citrate synthase. And again, here at 137 days of age, we see this almost threefold increase of enzyme expression here as well. So this tells us that there's something going on, and we're kind of narrowing in on this time point. At this point in pigs' lives, there are these major enzymatic changes that are occurring to support some sort of biological process that's happening in the muscle here. And then lastly, this moves us into the enzyme that takes us into an amino acid synthesis pathway. And if we look at muscle, you can see that between LD and MS, the more oxidative the muscle, the greater expression of GOT2, or this amino acid synthesizing enzyme. And again, at 137 days, so going from weaning to 137 days of age, we see this quite drastic increase in GOT2 expression here as well. And so if we look at all of these things together and we kind of bring this back to the growth curve of an animal, we know that when animals are growing, so from birth to weaning, we don't see a lot of growth happening here. But from weaning to market weight, there is this rapid and massive amount of tissue growth. And if we look at this broken down by tissue, so again, bone growth, where we're not going to want to intervene here. We need a strong skeletal system. And this really draws our attention to muscle growth. So a lot of the energy and nutrients during the animal's growth here are being diverted to muscle growth. And so as we kind of reach this point here where animals are plateauing or peaking in muscle growth, we seem to see these large enzymatic changes. So these changes of enzyme expression that are facilitating large amounts of protein accretion. And so this work kind of is the first to actually map out these enzymatic changes that are happening. And so we hope to use this to be able to make interventions, either identifying, using these enzymes that we've identified as genetic selection markers, or using these time points that we've identified as great time points to be able to focus in on nutritional advancements. And so this leads us to what we're going to be doing in the future and how we'll be building off of this research. So we'll be continuing to investigate what controls how these enzymes are responding. So we see that there is these increase in enzyme abundance at this time, but we still don't know what's facilitating that increase, and then what the importance of that increase is. So we'll plan to continue to investigate what's going on here. And then we're also going to start to look into these enzymes that we've identified. Can we use these as markers in breeding stock, or as animals are growing, as indicators of either keeping them in the herd as breeding stock, or maybe identifying pigs that seem to have a better or a little bit more primed to have greater muscle growth. So using these as markers, either for breeding stock or to separate them out and maybe expose them to different nutritional interventions. And that leads us into the last point of using these time periods that we've identified. So those last two time periods that we've identified seem to be very, very important to facilitate muscle growth. So maybe using this time point to change feeding, or do some further investigation here. And that leads us into the acknowledgements of how we've been able to complete this work. So first, I'd like to thank the graduate students that have done a lot of this work. So Linnea Rimmer has led a lot of the study, collected the samples, has done a lot of the lab work, and it also has been supported by Hope Oftenberg. She's another one of our master's students in the lab. And then the two undergraduate students that have been a part of this, Shanae Parker and Brittany Smith. I think that they both admit that they've learned a lot from this process, and actually now have an interest in attending graduate school in muscle biology or in swine science. And so having these funds had sparked the interest of these undergraduate students in research, in pig research. And then I'd also like to thank the rest of the meat science graduate students in the department. A lot of this was very labor intensive, so having the other meat science graduate students help out. And then the swine team here at K-State, this wouldn't have been possible without having the support from Dr. Woodworth and Goodband, who helped set up and facilitate the project as far as getting pigs and making this happening. And then lastly, I'd really like to thank the Kansas State Global Food Systems Seed Grant. This really allowed us the opportunity to start this work, and I think that we found some interesting findings that we'll then use in the future to continue this work and hopefully have a positive impact on the state.
Very good. Thank you, Dr. Zumbaugh. We would like to suggest if anyone listening has any questions or would like to discuss this research further, please feel free to reach out to Dr. Zumbaugh. Again, she is in the Department of Animal Science and Industry here at K-State, and I'm sure would be more than happy to talk with you about this.
Yes, thank you.
Thank you so much.